Good morning, guys. Chapter 19 is coming your way. So this is part 2 of the urinary system. But this one, we're going to focus much more on the functionality of this organ system. Besides producing urine, it's involving in maintaining, for example, water balance, maintaining electrolyte balance, and also involving in helping to maintain the pH in our blood plasma. So this is much more detailed in terms of physiology than part 1. And it is also much more interesting because of it. So let me kickstart the annotation here. Okay, so for this chapter, we're going to go over again understanding how our kidneys are maintaining a homeostatic balance of fluid, in this case water, and electrolytes, most importantly sodium. And amazingly, some types of cells, especially in the kidney cells, the renal cells, they spend up to half of all their cellular energy resources. Half of the ATP is dedicated to maintain this sodium balance. So this is an extremely important concept, because this concept comes back wherever salt goes, water follows, right? So this is the main mechanism by which the body is either trying to get rid of excess water or trying to retain water under cases of dehydration. So if you are not taking proper fluid intake, so you're not drinking enough water, then you're forcing your kidney to concentrate that urine. And that's on the menu today, talking about this fantastic mechanism that we call the countercurrent mechanism. And it is at the disposal of the kidneys to be able, allowing the kidney to concentrate, literally concentrate our urine to retain water under scenarios where you are being compromised, meaning you are being dehydrated. So the objective for the recording session is divided into 3 pillars. We're going to go over and understand how the body maintains water balance, followed by how the body maintains salt balance, focusing on putting an emphasis on sodium balance. And the last pillar is going over acid-base balance, how the body maintains the pH, specifically pH in our blood plasma. So those are the 3 pillars, those are the 3 learning objectives for the current recording session. So with that said, let's get the ball rolling. First is basically how we're focusing on the water balance here. Let me go back here briefly to reintroduce the idea. When we talk about urine production, remember we talk about 3 steps. We are talking about the glomerular filtration. And this glomerular filtration is a passive transport mechanism. It is driven by pressure gradient and is non-selective. So it's not selective. So anything that's smaller than the glomerular pore will go through, will leave the capillary bed and go into the Bowman's capsular space. So because of this non-specific passive transport mechanism through the glomerular membrane, what you find yourself in is not only all the waste will be filtered, but also water will be filtered. So you're losing water by this filtration process. You're also losing important nutrients. So for example, glucose, you know, glucose will be filtered. Amino acids will be filtered, right? So, and both the water and the nutrients and also important electrolytes, for example, sodium will also be filtered because they are so small, they will pass through the pore with no problem. So once you pass the filtration process, and all of this now find themselves in the renal filtrate. Right? And this renal filtrate will leave the renal corpuscle, meaning the Bowman's capsule with its glomerulus, and it will go to the PCT, proximal convoluted tubule. And it is exactly in the PCT that reabsorption of the most of the water, all of the glucose and amino acids, and most of the sodium would be reclaimed by the PCT. And because of that, we call this not absorption, but reabsorption, RE, and the word absorption, okay, here. Okay. So all of that desired material, water, nutrients, specifically glucose and amino acids, and sodium, and to some less extent, potassium, bicarbonate, or would be reclaimed by the proximal convoluted tubule. And the case of bicarbonate is a little bit special, and we'll talk about that bicarbonate later also, because that bicarbonate is a very, very important anion. It is involved helping, it is used as a buffer, preventing wild fluctuations in the pH in our body, whether it's in the renal filtrate, whether it's in the blood plasma. It is used to basically buffering acidification of our the pH in our body fluids. Okay, so that's basically bridging last recording to today's recording. So part 1 of chapter urinary system with part 2 today. Yeah. So water reabsorbed by at the PCT, proximal convoluted tubule, indirectly through also sodium reabsorption. So wherever salt goes, water just follows. Okay. And when we talk about water balance, there's a counter current mechanism. This is a very important mechanism. This counter current mechanism allows the kidney to create a steep gradient osmolarity gradient. Okay, a steep STEEP gradient gradient, osmolarity, osmolarity. And this deep osmolarity gradient occurs in the renal medulla. Okay. So in the kidney, you have the renal cortex, which is a superficial layer of tissue. And deep into the kidney, we call that region renal medulla. And in the regional medulla, it's a region, anatomical region within the kidney that contains all these renal pyramids. And within the renal pyramids, you have these extremely long loop of Henleys belonging to the juxtamedullary nephrons. So that renal medulla, starting from the renal cortex and going deep toward the renal pelvis, you have that osmolarity gradient. So as you get closer and closer to the renal cortex, the osmolarity is around closer to 300 milliosmol. That's the homeostatic uh, concentration of osmolarity, meaning concentration of all these electrolytes, uh, sodium salts mainly. And then as you go, you leave that renal cortex and you go deeper and deeper into the medulla, closer to the renal pelvis, then that gradient increases from 300 all the way to 1400, right? So that osmolarity gradient was created by this countercurrent mechanism. And that gradient, osmolarity gradient, is there just in case when the kidney needs to concentrate urine, right? So this is an important mechanism where the body or the kidneys can balance water, meaning it can concentrate urine when it is being dehydrated, the body is being dehydrated, or if the body is properly hydrated, then the kidneys produce dilute urine. So it can be producing dilute urine like 50 osmol all the way to 1400 milliosmol. So that's a wide range. And we're going to talk about that also in the animal kingdom, where some animal species can concentrate even further their urine. Camels, for example, they can concentrate twice as much as humans. And there's a species of mice where they can concentrate close to 10,000, to the point where they don't need to drink water. They can produce metabolic water, and they basically can survive with that water without drinking. Under cases where water is being compromised for intake, they just obtain the water from food, and we call that metabolic water. Unfortunately for us, we cannot survive on our own metabolic water. We don't produce enough metabolic water. So that's why we need to drink water. We need to have fluid intake. Okay. So let's go and explain this, learn this in detail of how this countercurrent mechanism works. And when we say countercurrent mechanism, there are actually two components. There's a counter current multiplier, which is here, counter current multiplier. And there's a counter current exchanger. The counter current multiplier, this is performed by the loop of Henle. Whereas the countercurrent exchanger, this is performed by the vasa recta. So it's a capillary bed that interacts closely with the loop of Henle. And when you hear about the word vasa recta, we are talking about juxtamedullary nephron with long loops of Henle, okay? Now, the countercurrent multiplier establishes that gradient, that osmolarity gradient, establishes the osmolarity gradient, whereas the countercurrent exchanger maintains that gradient. So that's a big difference, right? One mechanism creates the gradient, the other mechanism maintains that gradient, so prevents that gradient from dissipating, from disappearing. So the loop of Henle, the tubular system, creates the gradient. The capillary bed maintains that gradient. So they work as a team to basically allow the kidney to have that gradient in terms of osmolarity from the renal cortex all the way down to the renal medulla. So the entire medullary region of the kidney has a gradient of osmolarity. If you get closer to the renal cortex, we start at 300. And if you get close to the renal pelvis, the tip of the renal pyramid, then you're tipping at 1400 milliosmoles. Okay. And that gradient requires the interplay between the countercurrent multiplier and the countercurrent exchanger. And together they are known as a countercurrent mechanism. And we'll explain why we call it countercurrent. Okay. Let's get back here. So in your book, this slide, the second slide of the lecture outline, corresponds to two figures from your book, the Stanfield textbook. And that is picture figure 19.6 on page 538, and 19.7 on page 539, 539. So if you bring out your book and you follow the book, it should be easier for you. So again, one more time, the countercurrent multiplier, this is performed by the loop of Henle. And this loop of Henle is extremely long. The loop of Henle belongs to the juxtamedullary nephron, the nephron that basically can concentrate urine. There are only 15% of all the nephrons, so they are a minority. 85% of all nephrons are cortical nephrons. They have short loop of Henle. So we're not talking about the cortical nephrons here. We're talking about the juxtamedullary nephrons. Now, as you can see on the loop of Henle, there's two limbs, two parts. The descending limb, which is basically when you're coming out of the proximal convoluted tubule, so the renal filtrate comes from the proximal convoluted tubule, enters the loop of Henle through the descending limb, right? So that's the renal filtrate. So from the PCT, proximal convoluted tubule, the renal filtrate goes into the descending limb of the loop of Henle. And as the renal filtrate RF, travels through that descending loop of Henle, and this is where you see it on figure set 19.7 on panel 2. As it goes down, water is leaving, right? Because that descending limb is permeable to water. However, the solutes, the chemicals like sodium, is not allowed to leave. So sodium chloride, for example, the table salt that is in your renal filtrate remains in the renal filtrate. Only water is allowed to leave the renal filtrate and goes into the interstitial space, right? This is the space between, it's outside of your loop of Henle. So as water is leaving and the renal filtrate is going deeper into the medulla, you will see that in the renal filtrate, in the interstitial space here, in fact, you are losing, you are basically concentrating that renal filtrate because that water is leaving the renal filtrate. So if you go to panel 3, that's exactly what happens, okay? So as water is leaving the descending limb, if you lose water, you're concentrating the solutes inside of your loop of Henle, the descending limb. And as you go back up in the ascending limb, is the opposite. Now water is no longer able to leave. Water is stuck inside the ascending limb of the loop of Henle. But what is important at the ascending limb is there's an active transport mechanism requiring energy. And this is the part where it basically creates that, it contributes to creating that osmolarity, is the ascending limb consumes ATP, consumes cellular energy to kick out sodium, chloride, and potassium. So it is actively sending out, so the potassium, the chloride, and the sodium is leaving the renal filtrate and gets into the interstitial space. And you see that on panel 4 of figure 19.7. So as the renal filtrate is going back up through the ascending loop and going toward the renal cortex again, you're seeing that renal filtrate, the osmolarity, the salt concentration will drop. And it will go all the way down to, it can go all the way down to 50. So it started off here at 300 at the beginning. The osmolarity, the renal filtrate is around 300. But when it's leaving the loop of Henle, it can go all the way down to 50. Okay. In your book, it says 100, but it can go all the way down to 50. So this is how the loop of Henle is basically setting the concentration gradient, the osmolarity gradient, the osmotic gradient in your medulla. And that's panel 7 on the figure 19.7. Again, one more time, as the renal filtrate goes through the loop of Henle, on the descending limb, water is leaving. As water is leaving, the renal filtrate in the descending limb becomes more concentrated in terms of salt. That is going from the beginning of the descending limb closer to the renal cortex, and it gets deeper and deeper into the renal medulla. When you get to the loop, the helping loop of the Henle, it can go all the way to 1400 in terms of osmolarity, in terms of salt concentration. And when that renal filtrate goes back up through the ascending liminal loop of Henle, now water cannot escape, but that ascending limb is actively sending out sodium, potassium, and chloride. So that renal filtrate in the ascending limb is losing solute, is losing salt. Therefore, that renal filtrate in that ascending portion becomes more dilute. So that osmolarity will drop, moving from a deeper region of the renal medulla and going up toward the renal cortex again. Now you're seeing starting at 1400 and it's decreasing all the way to 100 as it's leaving that loop of Henle and it gets into the distal convoluted tubule, right? So here around this area, we are now moving into the DCT. We are leaving that loop of Henle. So as you can see here at the beginning, when it's leaving the proximal convoluted tubule, it has 300 milliosmol, the renal filtrate. And by the time it gets to the DCT, that renal filtrate can be down to 50 in terms of milliosmol. And when it becomes urine, we're talking of dilute urine, right? So in this case here, even though the osmotic gradient is present, it's not being used yet to concentrate urine. This is a tool that is readily there, and then we'll go on later to see how the body utilizes this osmotic gradient that is present in our renal medulla to concentrate urine. There's one more thing in addition to this, and surprisingly, the kidneys relies on a waste product when we catabolize our protein and we generate nitrogen. And the main source for nitrogen disposal is in the form of urea. So we basically excrete, waste product coming from protein breakdown in the form of urea. But we don't excrete 100% urea. We only, by the way, excrete only 40%. We reclaim 60% of the urea because that urea helps to maintain this osmotic gradient along the renal medulla. Because we have, urea transporters along that in both the tubular system and in the capillary. If you go to page 540, figure 19.8, you will see on that picture UTA, UTB, and UTC. UT stands for urea transporter, okay? Even in the collecting duct where you have urine, urea can leave the urine, leave the collecting duct, and move back into the renal medulla through the UTA, urea transporter type A. In the renal medulla, there's another type of urea transporter type B, UTB, will allow urea to move from the interstitial space in the renal medulla, and it gets into the descending limb of the vasa recta, which is a capillary, blood capillary. So urea, it gets in there and goes up through the ascending limb, but when it ends in the ascending limb of the vasa recta, it's not allowed to leave. Because of this, if you look at the vasa recta, when it's leaving, the blood is slightly more osmotic than the entry. So the blood gets in at the beginning of the vasa recta at 300 milliosmol, but when that blood is leaving the vasa recta, it's slightly more, it's around 325. This is due to urea, getting into the vasa recta through UTB. Okay. Urea can also sneak back into the proximal convoluted tubule from the interstitial space. And that urea transporter is UTC. So on the same picture, 19.8, you see UTC on the top of the picture. That is allowing the proximal convoluted tubule to absorb urea back into the PCT. So basically, urea is being recycled by the tubular system and by the vascular system. So 60% of that urea is being recycled. Only 40% is being disposed in urine. So moving on to the picture. This picture here is the equivalent. This part here you have the equivalent on figure 19.7, page 539, and 1910 on page 541 in the Stanfield textbook. Let me explain this. I talked about this, but this is the picture here. Going back again, because this is the best way to learn is through repetition. So you have here, this is the loop of Henle. And what we're talking about here, this is the long loop of Henle. This is the loop of Henle belonging to the juxtamedullary nephron, right? This loop of Henle dips down to the entire thickness of the renal medulla inside the renal pyramid, right? So the entire renal pyramid is located in the renal medulla. So that loop of Henle is extremely long. So the black arrows are basically showing you the renal filtrate. So you have here, blood plasma goes in here. That's the glomerulus. Blood plasma is filtered, and the black arrow comes out. That's the renal filtrate. So it goes from the renal corpuscle, it's leaving the renal corpuscle. It goes into the proximal convoluted tubule here, PCT. And as you can see, it's 300, 300, 300. That's the salt concentration. And it is expressed as milliosmol. Lowercase m, uppercase o, lowercase s, lowercase m. MOSM, milliosmol, 300. So this is at the beginning of the loop of Henle. So this is the descending limb of the loop of Henle. Because the renal filtrate is going down from the renal cortex, it moves into the renal medulla. So again here, this is your renal cortex. And this is your renal medulla. Okay? Now, as it goes down, the loop of Henle, you can see that renal filtrate gets concentrated. Why? Is because of this. Water is leaving. So basically, you are concentrating the renal filtrate. So the descending limb of the loop of Henle is concentrating that filtrate. That is basically moving deeper and deeper into the inner renal medulla. So we are concentrating from 300 all the way down to 1200. It can go as low as high as 1400. So anywhere from 1200 to 1400 milliosmol. We cannot, in human kidneys, we cannot concentrate more than 1400. That's the top that we can go, is 1400. So having said that, I'm going to erase this and erase this for clarity. And then the renal filtrate now goes back up. So this is the ascending limb of the loop of Henle. So that renal filtrate goes back toward the renal cortex, okay? Goes toward the distal convoluted tubule here. So this is the DCT here. So as that renal filtrate is moving back up, water is not allowed to leave. Water is not allowed to go in the ascending limb, or water is not allowed to leave the ascending limb of the loop of Henle. So that ascending limb is completely impermeable to water. However, it is actively sending out sodium, potassium, and chloride. So as the renal filtrate is moving up, that renal filtrate is losing salt. So that's why that renal filtrate is basically being diluted out again. So it's moving from 1200 all the way to 100 here. It can go as low as 50 milliosmol. And that renal filtrate can become dilute urine. In this case, 100 here, it's considered to be dilute urine. Okay. So anywhere from 50 to 100, that's dilute urine, meaning that urine is clear, looks like water. So that's a sign when you're basically urinating, you're peeing, and your urine is clear, then it's a sign that you know, okay, you are properly hydrated. Because if it starts to become yellow, we have a problem. We need fluid intake, okay? So that's the countercurrent mechanism. This is the loop of Henle establishing the osmotic gradient along the renal medulla, right? So the entire thickness, entire region of the medulla, along that medulla, you have a gradient starting from around 300, close to the cortex, and all the way down to the inner medulla at 12 to 1400 milliosmol. And in order for this to occur, the only reason why the countercurrent mechanism, the countercurrent multiplier, meaning the loop of Henle can do that, is because of this. I need to erase this. This part here, active transport. The ascending limb is setting the tone. The ascending limb loop of Henle consumes energy, consumes ATP to actively, okay, sending out sodium, potassium, and chloride, allowing them to leave the renal filtrate and go into the interstitial space. The space that is basically between outside of loop Henle and outside of vasa recta. It's just that space outside of the vessels, outside the cells, okay? And that is what sets the tone. That step is the actual step that basically the beginning to create that osmotic gradient from the renal cortex, the outer medulla, to the renal inner medulla. So throughout that medulla, renal medulla, that osmotic gradient is possible because of the ascending limb of the loop, ascending limb of the loop of Henle, using active transport to actively pumping out salt, concentrating that renal filtrate as it goes toward the DCT. And that step, we call it countercurrent multiplier. So the countercurrent multiplier establishes the gradient, this gradient here. When we say osmotic gradient, that's the gradient we're talking about. And this is in the renal medulla. And it can go not only 1200, it can go up to 1400. Okay, so that's osmotic gradient. It is set, it is created, it is established by the loop of Henle, more specifically the ascending loop of Henle, by relying on active transport. If ATP runs out, this is off. There's no more osmotic gradient. Okay, so that osmotic gradient requires a constant input of energy. Okay, so that's the counter current multiplier, setting it up. But we need something else. We need another mechanism. It's called a counter current exchanger. And the counter current exchanger is going to be on figure 19.8 and 19.9 of your Stanfield textbook. And we are specifically referring to the vasa recta. This is the capillary bed that is exclusively, intimately interacting, physically interacting with the long loops of Henle of the juxtamedullary nephron. So when you hear the word vasa recta, you know that they are basically interacting with these extremely long loop of Henle in the renal medulla, okay? Because the shorter loop of Henle of the cortical nephrons, they also interact with the capillary bed, but that capillary bed is not vasa recta. That capillary bed is called peritubular capillaries. So those cortical nephrons, they always produce dilute urine. They don't have that osmotic gradient. And their loop of Henle is not deeping into the renal medulla, right? So because of that, they don't have that osmotic gradient, so they cannot produce concentrate urine. They can only produce dilute urine. So when vasa recta, that's the counter current exchanger. The vasa recta, maintains that osmotic gradient. Okay, so let me go back to the picture here. I don't think the next slide is, yeah. So it's the same picture. So this one here. Now this is the vasa recta. Now why do we call it counter current? If you look on the loop of Henle, the renal filtrate goes into opposite direction. The descending limb, the renal filtrate is going away from the renal cortex. The ascending limb, the renal filtrate goes in the opposite direction. It goes toward the renal cortex. So counter means opposite direction. Same thing with the vasa recta. The descending limb of the vasa recta also goes down toward the loop, toward the tip of the vasa recta. And the ascending limb, it goes up in the opposite direction, going back to the vein here. Okay. Now, what is important is, and in your book, it's clear if you look and go to the figure 19.8 on page 540, you see the loop of Henle as a yellow structure, the hairpin loop, yellow hairpin loop, and the vasa recta is the red colored vessels and blue colored vessels. If you pay attention to the arrows, right, the descending limb of the loop of Henle, the yellow, the black arrow is going down, but the blood vessels next to it, the blue blood vessel, the ascending limb of the vasa recta, blood is going up. So even the interaction between the loop of Henle and the vasa recta, they have opposite flow. In the loop of Henle, we're talking about the flow of the renal filtrate. In the vasa recta, we're talking about the flow of blood. So blood plasma flow, right? Or blood flow. So the ascending limb of the loop of Henle interacts with the ascending limb of the vasa recta, opposite flow. The ascending limb of the loop of Henle interacts with the descending limb of the vasa recta, again, opposite flow. So let me draw it out here. So you have here, this is the descending limb of the loop of Henle. This part is interacting with this part. Okay, blue with blue. And the red part, the ascending limb, is interacting with this part. Okay. So if you look closely, this black arrow is going down for the blue part. The blue part here, arrow is going up. So this is going down. And this is going up. Okay. Oh, I should have done it in the same color. It's back to blue. Again, this is going down. Descending limb of the loop of Henle. Ascending limb of the vasa recta going up. Opposite direction, counter current. For this part, ascending limb renal filtrate goes up. For the vasa recta, it goes down. Right, so opposite direction for red, opposite direction for blue. So if you, again, if you look close, I'm going to erase this for clarity. Okay. So if you see here, all this water, when it's leaving, it needs to go somewhere. Otherwise, it's going to dissipate, right? So this water, when it's leaving the descending limb of loop of Henle, it will be received over here. So water will go into the ascending limb of the vasa recta. Same thing over here. When sodium is leaving the ascending limb, that sodium is allowed to go in the vasa recta. So by doing that, both the vasa recta and the renal, the loop of Henle, have the same osmotic gradient, as well as the same osmotic gradient interstitial space, right? So if you look closely, that osmotic gradient in the interstitial space here, the yellow with the red line rectangle, it's the same, and you look closely with the loop of Henle and with the vasa recta, right? At the bottom here, it can go up to 1400, 1400, and 1400. And urea, like I mentioned earlier, helps to maintain that as well. So urea is reclaimed, can be reclaimed from the collecting duct through urea transporter being inserted into the cell membrane of these cells, lining along the wall of the collecting duct, allowing urea to get it back in. So urea would leave the urine and get back into the renal medulla. So urea is being recycled to maintain that osmotic gradient. So the vasa recta prevents the osmotic gradient from dissipating. So that's why it's known as the one that maintains that gradient, right? So the countercurrent multiplier, the loop of Henle, utilizes cellular energy, ATP, to set up that osmotic gradient. But the vasa recta, the countercurrent exchanger, helps the countercurrent multiplier to maintain it. Okay. So once you have that, the renal medulla has this osmotic gradient. Then the kidneys can use that gradient to concentrate urine. At this point, it hasn't used it yet. It's just even the juxtamedullary nephron is producing dilute urine. Even though the gradient is there, the osmotic gradient is present 24/7, but the juxtamedullary nephron is keeps producing dilute urine if the body is properly hydrated. So in this case, both the cortical nephron and juxtamedullary nephron produce dilute urine if you have proper fluid intake. Now, in the case where your body is being compromised, being dehydrated, so what's going to happen is the posterior pituitary gland, which stores this ADH, antidiuretic hormone, which was produced by the hypothalamus, by the way, but is being stored in the posterior pituitary. Okay, so this is a hormone, antidiuretic or antidiuretic hormone. Okay. So when you have dilute urine, meaning when you are properly hydrated, ADH is not released by the posterior lobe of the pituitary gland. PG is in posterior pituitary gland. So you can say posterior lobe of the pituitary gland or simply posterior pituitary. Okay. So when ADH is not released, the kidneys keeps producing dilute urine, anywhere from 50 milliosmol to 100 milliosmol. Both cortical nephrons and juxtamedullary nephron are basically doing that, just that. Aquaporins, those are important. Those are transmembrane proteins that are inserted into the plasma membrane of the cells that line the walls of the collecting duct. So that's why we say PM stands for plasma membrane, CD collecting duct cells, right? So the plasma membrane of these cells will receive these aquaporins. Aqua means water, porins means pores. So these are water pores that gets inserted into the plasma membrane and allows water to leave urine, right? We're talking about a collecting duct here. So the water in your urine will leave the collecting duct through the aquaporins and get back into the renal medulla. Okay? And you see that here. So if you go to, this is from your book, figure 1910. So let's start with this, panel A. This is where you are properly hydrated. So under this case here, proper hydration, pH, right? Proper, P-R-O-P-E-R, hydration, H-Y-D-R-A-T-I-O-N. So if you are properly hydrated, the urine is dilute. Nothing happens. But if you are dehydrated, this is what happens. So the collecting duct now have aquaporins inserted so water can leave, right? So this ADH is released by the posterior pituitary, and the ADH induces the collecting duct cells to insert aquaporin into the plasma membrane, allowing water to leave the collecting duct. And you have a picture in your book about that. And that picture here specifically is 19.11 on page 543, right? So you see on that picture, the ADH is a hormone released by the posterior pituitary gland. It's leaving the capillary bed, and it goes into the receptor. There's an ADH receptor, which activates the G protein, and then the rest is history. We learn about that. Ultimately, activating protein kinase A, and that PKA, protein kinase A, induces the insertion of aquaporin number 2 onto the plasma membrane, onto the apical membrane of those cells. And those cells are called principal cells that are lining the wall of the collecting duct. You have on the basolateral side of the same principal cells, aquaporin 3. That aquaporin 3 is always there. Aquaporin 2 at the apical membrane is not there unless ADH induces them to be there, okay? So ADH has a direct influence on the insertion of aquaporin 2 on the apical membrane of the principal cells lining the collecting duct, allowing the body, allowing the kidneys to reclaim water, because the body needs water. The body is being dehydrated. So that's why you are concentrating your urine. Now, there's a limit as to how much you can concentrate urine. So that limit is you cannot concentrate urine more concentrated than your own osmotic gradient in the renal medulla. So if the osmotic gradient is 1400, then the most concentrated urine you can produce is 1400. You cannot produce more concentrated urine than your own osmotic gradient. And because of that, we always need a minimal amount of water to basically excrete our waste. And that minimum amount of water is called obligatory water loss. Obligatory, OBLIGATORY, water loss. And it's slightly less than 500, half a liter per day. More specific, around 440 to 450 ml per day, that you cannot keep, you have to lose, because that water is the minimal amount of water, the least amount of volume required for you to basically excrete your waste, right? So you will lose slightly less than half a liter per day, at least, just to eliminate your cellular waste. Okay. So that would complete the countercurrent mechanism using the countercurrent multiplier and then countercurrent exchanger to establish and maintain that osmotic gradient throughout the renal medulla. And when the body needs it, it releases ADH, and ADH induces the insertion of water pores into the plasma membrane of the cells lining the wall of the collecting duct, allowing water to leave the urine and get into these cells, and ultimately leave these cells and get into the body. So it allows the body to literally concentrate that urine, to keep water, to retain water, because the body needs that water, because the body is being dehydrated. So this is the central role of sodium. So that ECF stands for extracellular fluid. So most of it is sodium salt, up to 95% of it. So majority of it in the form of two types of sodium salt, sodium carbonate and sodium chloride. Sodium chloride is a table salt, right? So out of 300 milliosmol, the sodium salt contribute 280. So more than 90% of that osmolarity is coming from due to the presence of sodium salts in our extracellular fluid. And the body regulates sodium balance by using a steroid hormone, mineralocorticoids, that we saw during the endocrine system. Aldosterone is being produced by the renal cortex. Like all steroid hormones, you have only two anatomical locations where you can produce them: adrenal cortex and the gonads. So aldosterone is the most important mineralocorticoids involved in maintaining sodium balance, right? So regulating, allowing the kidneys to contribute to regulate sodium in the extracellular fluid. And these cells in the kidneys spend half of their energy resource, half of the cellular energy or ATP supply, just to maintain that salt balance. Why? Because that salt is indirectly influencing water balance. That statement comes back. Wherever salt goes, water follows, right? If you want to keep water, keep salt. If you want to get rid of water, get rid of the salt, okay? And this aldosterone, we talked about this already, but let's revisit this. This is also known as the renin-angiotensin pathway. So it is basically related to the release of angiotensin 2. So let's go and briefly talk about this on the next slide. So what you have is angiotensinogen is a glycoprotein, a protein with a sugar groups attached to it, and is produced by the liver, and it belongs to this is an example, a specific example of plasma protein. We have three families of plasma proteins, right? From anatomy: albumins, globulins, and fibrinogen. The angiotensinogen belongs to the class 2. It's grouped under the globulins family. And that angiotensinogen floats, in our blood plasma. And the juxtaglomerular cells in the kidneys release an enzyme, renin. And this renin will cut angiotensinogen on the N-terminus of the angiotensinogen, the protein, at the beginning of the protein, generating angiotensin 1. So this is A1, angiotensin 1. And angiotensin 1 is inactive. And on the endothelial cells, meaning the simple squamous epithelial cells lining the capillary walls, they produce and release another enzyme called ACE, angiotensin converting enzyme. The word angiotensin converting enzyme. And this enzyme will convert angiotensin 1 into an active hormone, angiotensin 2. This is one of the most, if not the most, if not the most, one of the most powerful vasoconstrictor. It will cause constriction of your arterioles. If it constrict, it will increase blood pressure, meaning it will increase TPR, total peripheral resistance. And if that's the case, then it will increase MAP, right? So MAP, again, is the driving force pushing blood through your blood vessels. MAP equals the heart rate times the stroke volume times TPR, total peripheral resistance. This here is cardiac output. This is one of the questions from the previous exam. So the question was asking you, ignore cardiac output. And what would be the consequence of, norepinephrine, right? Acting on alpha adrenergic receptor. So if you ignore cardiac output, the only way that it can affect is TPR. So if it binds and then it causes constriction, it will increase TPR. If it increases TPR, then it will increase MAP. Right? So that's the connection that you will require to understand. This is critical thinking here. So the question is asking you, it tells you, ignore cardiac output. Meaning, you can ignore, according to the question, HR and SV is not contributing. So how norepinephrine can affect MAP? Well, the only way is through TPR. So norepinephrine will cause vasoconstriction, increasing TPR, therefore increasing MAP. It's a gorgeous question, right? Okay, let's get back here. I lost my line of thought. Okay, so angiotensin 2. Now we take it to another level. We stopped there in the previous unit, but now this unit, angiotensin 2, in addition to causing vasoconstriction, it will induce the adrenal cortex to release aldosterone. And this is the mechanism of aldosterone here. So aldosterone will basically go, this is a steroid hormone, so it's fat soluble, it goes straight into the cell. There's a receptor here. So when aldosterone binds to its receptor, it will basically induce more protein synthesis. So it induces the cell to produce more sodium channels, more potassium channels, and it increases the flux of sodium and potassium, right? It also activates the potassium and sodium pumps down here. So basically it's creating the gradients. So by sequestering sodium away, the intracellular consumption of sodium lowers, so it induces sodium to get in, and it kicks out potassium, right? So here, the cell is conserving sodium, but kicking out potassium, okay? Principal cells, and this is depending, you can be the distal convoluted tubule or the collecting duct. So if we are talking about the DCT, then we are talking about the renal filtrate. But if we are talking about the collecting duct, then we are talking about urine. This compartment here, okay? So when it says tubular fluid, depends on which region of the tubule system. Are we talking about the DCT or are we talking about the collecting duct? So this is how aldosterone is basically helping the body to conserve water by bringing in sodium. If sodium gets in, water just follows. I'm looking for a passage in your book where it tells specifically the 4 functions of angiotensinogen. Yeah, I found it. Okay. In your book, on page 546, write the part where it says figure 19.17. Underneath that, it has the number 1, 2, 3, 4. Those are the 4 functions of angiotensinogen 2. It is required that you go over that and you learn it as is. It's very detailed instead of me repeating myself. But it tells you the story that I just told you. So what are the functions in addition to the fact that angiotensinogen is causing vasoconstriction? And it tells you the story in writing. So you have that in addition to what I explained earlier. You can have that in writing. It's on page 546 of your Stanfield textbook. Moving on to chemical buffers. The chemical buffer system is really quick to basically try to prevent fluctuation of pH. So we have 3 systems. We have the bicarbonate buffer system. We have the phosphate buffer system. And we have the proteins can also act as a buffer. Bicarbonate is the main buffer system for extracellular fluid. That fluid is outside of your cells and outside of vessels, right? I take that back. Extracellular fluids means only the fluid is outside your cells. Phosphate system is used in your urine and intracellular fluid inside your cells. And the protein buffer system is intracellular fluid and blood plasma. Huh? Now the chemical buffer systems are fast, but they can only prevent it to some certain extent. What they cannot do is basically get rid of the protons. So they cannot eliminate completely protons, free-floating protons that affects pH, right? The chemical buffer system cannot do it. It's just a quick fix. It's just basically shuffling that proton around. The protons is basically still inside the system. So the chemical buffer system cannot do that. Moving on to the next one, the respiratory system is also fast, also quick, but we have the same problem. It cannot get rid of those protons. Okay? And the respiratory system is our lungs. The way you breathe, you can basically changing quickly the pH of your blood plasma by the same relationship. This relationship here, this is one of the most important equation for your entire clinical career. The relationship between CO2 and proton, because this proton is related to pH. Free-floating protons. So that's why the body never allows proton to freely float, because if you allow proton to freely float in a solution, it will acidify your solution. Your solution, your pH, the pH of your solution will drop, right? And because of that, just by the fact that you're breathing in and out, and this is equilibrium here, this should have been written like this. It goes this way. It can also go in the opposite direction. Same thing here. It can go this way, or it can go this way. Okay, so those are two equilibria. So let's go slowly over this. This is chemistry, so I just want to make sure that we're on the same page here. This is basic chemistry. This is a law. So this side is the reactant, right? So if the concentration of CO2 is increased, you will drive the equilibria this way. So the equilibrium maintains the concentration. So if this CO2 is increased, these two equilibria will try to decrease it by moving excessive CO2 that side. So what's going to create? It's going to create proton indirectly will also then increase. And if this proton increase, it will lead to acidification of the pH of the solution. So pH will drop. Okay. So if you increase CO2, you acidify your solution. So how do you increase CO2? Meaning CO2 is being accumulated in your system. That happens when you are hypoventilating. So you're not breathing properly. So when you're not breathing properly, CO2 in your blood plasma increases, leading to an accumulation of protons, free floating protons. It will acidify your blood pH. pH drops. We call that respiratory acidosis. Okay. Now let me reverse the process now. What if instead of increasing, we talk about decreasing? If you decrease CO2, the equilibrium will try to bring it back. So it's going to be pushed in this direction to bring it back up. So if that's the case, then proton will also drop. So free floating proton drop, pH will increase. So when CO2 drop is when you are hyperventilating. You're breathing at a high rate, right? Hyperventilating. And when that happens, you have respiratory alkalosis. And remember, the value of the pH is highly regulated, is 7.35 to 7.45. Okay, higher than 7.45 is alkalosis, ALK. If it's lower than 7.35, it's acidosis, ACI. Okay. Now, what's the difference between the two? The chemical buffer system versus the respiratory system. Let me just erase this first. The Respiratory regulation of proton belongs to what is called a physiological buffering system versus the previous one, which is chemical. So we have chemical buffer system and we have physiological buffer system. The physiological buffering system, especially the respiratory regulation, is slower acting than chemical buffer system, but it's more powerful. And this is here, this is amazing. Just by increasing breathing rate twofold, you are creating a change in 0.2 pH. That's basically outside of your normal range anyway. So that's how powerful it is. Or you just drop your breathing rate half and you will also cause a 0.2 pH change. Okay. So this is much more efficient than the chemical buffer system. However, both of them, both the chemical and this respiratory regulation, they cannot get rid of protons. The proton is still inside the body. So even though they are fast, powerful, whatever you name it, they cannot get getting rid of the problem. Okay, accumulating proton is still in the body. Sooner or later, that accumulation will oversaturate both the chemical buffer and the respiratory buffer system. So both the chemical buffers and your lungs will no longer be able to deal with that much proton in the system. It keeps accumulating, so the problem has not been solved. It's just a quick fix, so we need something else. And that something else, the star of the night, is our kidneys. They are the third mechanism. They are also fall under physiological buffer system, but now we're not talking about the lungs, we are talking about the kidneys. And the kidneys can and will get rid of these protons. They are the slowest onset, they take time to kickstart, but then they can get rid of our metabolic acids, meaning acids that are produced by, metabolic pathways. So as our cells produces, using chemical reactions to obtain energy, they produce side waste products, and a lot of them are acidic, and we need to get rid of them. And the only way is through urinary excretion. Okay, which leads us to basically the last two slides. So the renal mechanism of acid-base balance. So metabolic acids generated by cells or acids generated by cellular metabolism. You have phosphoric acid, uric acid, lactic acid, ketone bodies. Lungs, again, cannot dispose these acids. Chemical buffers cannot dispose these acids. Only the kidneys can, okay? Now, the kidneys can also maintain the alkaline reserve, meaning bicarbonate. So they have two ways. They can reabsorb the filtered bicarbonate, and ultimately it's not enough. The body needs more than that. So what is amazing is the kidney also can generating new bicarbonate. And this is an amazing ability, because that bicarbonate is used as a buffer. to prevent acidification of our body fluids, and also to neutralize that gastric juice coming from your stomach into your small intestine. So when your chyme is moving from your stomach into your duodenum, the first part of the small intestine, the main job, the first job of the duodenum is to neutralize that gastric acidic, chyme. It will bring it from anywhere from pH 1 to 2 all the way to pH 8, just by secreting bicarbonate into the digestive juice coming from the pancreas. So the pancreatic cells produce this digestive juice containing all these enzymes, to cut proteins, to cut lipids, but also containing bicarbonate. That job is to neutralize that gastric acidity and bring it to pH 8. Why pH 8? Because most of the digestive enzyme in the small intestine, the optimal pH is pH 8. Right? So bring it to pH 8 so they can function optimally. And that's one of the functions of bicarbonate. So that's why we need lots and lots of bicarbonate. So reabsorbing filtered bicarbonate is not going to be enough. We need to produce new bicarbonate, and that the kidney cells will do. In addition to filtering and producing urine, in addition to maintaining long-term blood pressure, now you're seeing they can generate new bicarbonate to help maintaining acid-base balance. So we take for granted our kidneys, but they are an amazing waste management system. There's also the concept of losing bicarbonate. So the body can also secrete that bicarbonate, but that's not the predominant process. The main process is bicarbonate reabsorption. And amazingly, to this point, to this day, the exact mechanism as to how the body is secreting bicarbonate is still unclear. So the exact molecular mechanism is still unknown. So that's another topic for your PhD thesis if you want to do, pursue your PhD degree, becoming a scientist. Okay. So with that said, let's go over and see how all of this is panning out. How do we reabsorb filtered bicarbonate, and how do we generate new bicarbonate? So here, conserving or recycling filtered bicarbonates. Again, this bicarbonate was filtered by the glomeruli. So this bicarbonate was filtered from the glomerulus and is finding themselves into the renal filtrate, and that needs to be reclaimed because that's a desired chemical. Okay. So to maintain that alkaline reserve, and this is describing it. So it happens, it can be done by the proximal convoluted tubule. It can be done at the collecting duct. And if it's at the collecting duct, then it's a type A intercalated cells of the collecting duct. Now we have the problem. Bicarbonate is water soluble. It cannot cross cell membranes. It cannot cross plasma membranes. So we have to have a trick, right? Since all cells are impermeable to bicarbonate, this is water soluble. Okay. So let's go over here on the next slide and see, this is a nice trick. It's not really fancy, but it works. Now, you have bicarbonate here, and this is at the PCT. So we are talking about the proximal convoluted tubule, right? So PCT cells. So what you have is, this was filtered. So this was filtered out. So this is the renal filtrate here, coming from the renal corpuscle. So this was filtered out. So bicarbonate cannot get in. So what bicarbonate can do is basically it can reconvert back to CO2. This chemical reaction here is here. The two equilibria we talked earlier. Okay? So it reconverts back to CO2, and now CO2, we learned from a previous recording, it is highly soluble. It can go across the cell membrane. It is much more soluble than oxygen. So it can also cross the cell membrane here. And when it gets in CO2, it reconverts back to bicarbonate. And that bicarbonate will reclaim into this peritubular capillary. So you have this bicarbonate ultimately going in here. Okay. So that bicarbonate wasn't new. It's just reclaimed, reabsorbed. This bicarbonate was filtered. This bicarbonate here is reabsorbed, not absorbed, reabsorbed. Okay. Now the process is very simple. We learned all of this. So there's an ATPase pumping the proton out. Right? So if CO2 comes in and you convert CO2 to CO2 to bicarbonate, that proton needs to be kicked out by two mechanisms. There's a proton pump, ATPase, it pumps only proton. That's a primary active transport. So this ATPase only pumps proton out. Okay? There's another mechanism. This is the secondary active transport. This part here. Sodium gets in and proton gets out. The reason why sodium gets in is because of this. Sodium potassium ATPase. That mechanism we learned already. So this sodium potassium ATPase creates a concentration gradient of sodium, allowing sodium to get in and kicking out proton. So there are two ways for the proton to leave this PCT cell: by a primary active transport. So the proton ATPase pumps out only proton, or through a sodium proton symporter, co-transporter. No, sorry, I take that back, antiporter. Sodium gets in, proton gets out. Okay, so again, two ways for proton to leave the cell: through a primary active transport, its exclusive proton pump, consuming ATPase to pump out proton. And the second mechanism, it's a secondary active transport, where proton is basically gets a free ride out as sodium gets in. And why is that sodium getting in? Is because at the basolateral membrane, the sodium potassium ATPase sets up that concentration gradient, inducing sodium to move from the renal filtrate into that PCT cells. So this is how bicarbonate gets reabsorbed. And this is like a stone striking two birds, one stone striking two birds. Not only you reclaim bicarbonate, but you are also, and this is important, you're also getting rid of the proton, right? So the proton here gets out, but the chemical buffer system cannot do this. The respiratory system cannot do this. Only, again, only the kidneys can, okay? So not only you're reabsorbing something you desired, but also you're getting rid of something that's undesired. So you're reclaiming bicarbonate, you're getting rid of these hydrogen cations, which affects directly the pH of your body fluids. Now, that is just reabsorbing what you have. And we know it's not enough, right? So we need to produce new bicarbonate, which leads to the next slide. So there are two mechanisms allowing the kidneys to produce new bicarbonate. The first mechanism is this one. So let's go over that one first, which is this picture here. It corresponds to figure 1927 in your book. Let me go there. So 19.27 is page 558 in your book. Oh, the previous picture, sorry, I forgot. This picture here corresponds to 1926 of your book, and that's on page 557. Okay. Let's go back here. This one. Now, in this case here, it's called a buffered proton mechanism. So what you have is the cell is producing its own CO2. So every cell does that, right? So aerobic cell respiration, as you're breaking down glucose through glycolysis, through Krebs cycle, and through electron transport chain, you're generating CO2. And in this case, some of this CO2 is utilized to generate bicarbonate. And that proton gets kicked out. Again, this is primary active transport. This is a unique proton pump consuming energy to kick out protons. Now, this proton will acidify. This is the collecting duct. So this is basically, if it's collecting duct, then this should be urine, right? So we learned earlier, one of the chemical buffer in urine is the phosphate buffer. So phosphate is there. Phosphate is also filtered from the glomeruli. So this was also filtered out. And it will being used as a buffer. So this phosphate binds with the proton, preventing there's no free-floating proton. And this is the buffer here. So this is the reason why urine, it has a pH around 5. So it's slightly acidic, right? But without the buffer, without phosphate, that urine, our urine will be pH 1. And that means death. That pH 1 will puncture every cell, will burn your kidneys, right? So to prevent that from happening, those free-floating protons will be bound by a phosphate salt, and it basically neutralizes, bring pH 1 to 2 to pH 5. So this is why urine is slightly acidic. So that's one way to produce new bicarbonate. However, under severe acidosis that requires lots and lots of bicarbonate, none of the mechanism we saw up to now is going to work, including this one. The buffered proton mechanism is not enough. Our last hope is the last mechanism. This one here, the glutamine metabolism. Okay. The previous mechanism, type A of collecting duct, proton is secreted via proton ATPase. This is a primary active transport. Proton hydrogen or potassium proton antiporter. Okay. So this is the mechanism on this side of the picture. Now we're going to focus on this one. So this is the most important mechanism, allowing the body to create new bicarbonate. And this is how it works. This glutamine is an amino acid. And that amino acid was also filtered by the glomeruli, right? So it was filtered out. And we're talking about PCT here. So this is the renal filtrate. So as glutamine gets in, amino acid gets in, it will be basically, each glutamine will be used to produce 2 bicarbonate. Ammonium here gets out and excreted in the urine. Sodium gets in. The reason why sodium gets in is because of this, sodium potassium ATPase. So this drives sodium getting in, and ammonium gets a free ride being kicked out, and bicarbonate gets in. This is new. So for every single glutamine reabsorbed, it can generate 2 bicarbonate. This mechanism kicks in when we have severe acidosis, when our pH, the pH in our blood plasma drops significantly. This mechanism kicks in. If the kidneys fail to provide the glutamine metabolism, then the patient will die due to metabolic acidosis. Okay. Now, a question I know that you can answer: what would be the mechanism of transport of glutamine inside the cell? And that's a question for you. So if you spend some time on it, and if not, you cannot arrive at an answer, just ask me on your discussion board, okay? Hint, it is very, very, well, I can't give you the hint, because if you give you a hint, I give you the answer. But it involves one of the transport mechanism that you already learned, okay? So try to figure it out, try to connect the dot. And on that note, I will terminate today's recording, and I will see you on the next one. Take care, everybody. Stay safe, okay? Be happy, be healthy, be safe.